Thank you, Dr. Yamamoto. Thank you very much for your kind introduction. This is Takayuki Morisaki, BioBank Japan. And it is a great honor for me to have an opportunity to talk about BioBank Japan project in this symposium. My presentation title is here, the BioBank Japan, 20 years of disease-oriented cohort study towards genomic and multi-omic study. The BioBank Japan project started in 2003 for the implementation of personalized medicine. Now we are in the fourth phase of this project from the fiscal year 2018 and in the 20th year of this project. There are two types of biobank, population-based biobank and disease-oriented biobank. BioBank Japan is the latter, disease-oriented biobank. The participants were recruited in the collaborative hospitals, and the medical records are the source of clinical information, and we have the result of interview. 20 years of BioBank Japan. The first phase, as I said earlier, from 2003 to 2008. In this phase, the project recruited 200,000 patients. In total, there are about 300,000 cases with 47 common diseases, including several cancers and malignancies. DNA and serum samples were collected, and the medical record and interview record were also collected for the clinical information database. Until the end of second phase, 2013, the project continued, and the follow-up of these patients and collected in medical record and serum sample yearly. The third phase started in 2013. The project newly started to recruit additional 67,000 patients with 38 diseases, and that's about 100,000 cases, including common disease and cancers. Medical records were collected as in the first stage, but only the DNA and the clinical records were collected. In the fourth phase of this project, starting in 2018, the project improved the process of sample data provision. Also, based on the progress of genetic studies and metabolomic study, genetic data and some of metabolic data started to be provided to researchers with clinical data. In total, 267,000 patients, sample data of 440,000 cases with common diseases and cancers were being provided for researchers in this period. Here is the slide show the characteristics of BioBank Japan. Our project is one of the largest disease-oriented biobank in the world. Participating patients is about 267,000, as I said earlier. Our target diseases are totally 51 common diseases, including malignancy. We did long-term follow-up up to 10 years of participants. We are providing sample data to researchers in academia and the companies, though at the moment, individual sample data provision were for domestic researchers in Japan at the moment. Now, as you know, there are not a few biobanks and cohorts in the world. Slides show the global distribution of biobank and cohort, though not all of them are depicted here. This is the showing the BioBank Japan participants. Those patients were recruited in 12 cooperative medical institutes running more than 60 hospitals located through northern part to southern part of Japan, as shown here. As I showed earlier, in the first phase of the project, we recruited 200,000 patients with 47 diseases for 5 years. We called it as the first cohort. And then these individuals were followed up. In the third phase of this project, we recruited additional 67,000 patients with 38 diseases for 5 years. We called it the second cohort. In the first cohort, we collected DNA samples, serum, and clinical information, while we collected DNA and clinical information in the second cohort. Okay. The slide shows the diseases and the number of the patients recruited in the project. Totally, we collected samples and data as shown here. The most number of the patients have the dyslipidemia or diabetes mellitus, but several cancers like prostate cancer, gastric cancer, breast cancer, and so on. We are running 3 types of storage system called bank for collected samples. DNA bank is an automated storage system at 4 degrees Celsius, having capacity of 2 million sample tubes. That means the 2 units having the capacity of 1 million tubes. And currently, we have 800,000 tubes for our project. Our second bank is serum bank, consists of the 58 liquid nitrogen tanks having total capacity of 340,000 sample tubes at minus 150 degrees Celsius. In total, for our project, about 2 million tubes are being stored. Third bank is the tissue bank. This system consists of 12 units of automated liquid nitrogen tank system, having a total capacity of 480,000 sample tubes at minus 150 Celsius. For tissue bank, is not for BioBank Japan project, but for the clinical research groups all over Japan. So we have checked the quality of stored DNA samples since we are now in this 20th years from the start point of our project. We checked the stored DNA sample quality and found it fairly good quality. And also, we found that the result of simple array analysis showed a good call rate. Eventually, we obtained the foreign genomic result. That means the SNP typing data for all of 267,000 patients. And the first cohort is as shown here for the about 190,000 patients. Cancer panel sequencing data for 30,000 patients, and the whole genome sequencing data for about 10,000 patients, and more sequencing results are being collected. In summary, we have not a few numbers of samples and data for researchers. They include 267,000 DNA samples and serum samples of 200,000 patients. That means about 2 million tubes. Since we have multiple times a collection of serum samples yearly. Also, we clinical data information stored, and there are more than 5,000 items. Regarding genomic data, SNP data for all of 267,000 patients are finished, and about 220,000 patient data was available from the public database. Also, 30,000 cancer panel sequencing and some of the finished whole genome sequencing data for 10,000 patients. In addition, nowadays more than 48,000 metabolomic data will be available for researchers. About regarding the whole genome sequencing and metabolomic data, we are generating more results near future. Regarding genome phenotype data we obtained in this project, most of data have been submitted to the public database NVDC shown here in Japan. Researchers in Japan can access the data for their research purposes according to the specified processes. Regarding the first phase recruitment of patients, yearly taken serum samples and clinical follow-up data have been collected and provided for the researchers. Among the 200,000 patients, more than 800 patients with diabetes were followed up 10 years and have been 10 times collected serum samples. The information, especially those chronic diseases, for example, diabetes medicine, triopathies in the diabetes information collected, and those information and the data as well as the serum sample will be provided for the researchers. And for convenience of researchers, we have established two types of online sample search system. One is the BBJ online biological sample search system, and the other is made by the Tohoku Medical Megabank and BioBank cross-search system among several biobanks in Japan, including Tohoku Medical Megabank. Regarding the steps for provision of samples data, in the present fourth phase of the project, we have multi-step procedure for provision of samples data, including electrical radio system, resulting in shorter time of the sample data provision. First, researchers can consult on the access of samples data for our office, and then they should obtain IRB approval. Then they can make an application to the Samples and Data Access Committee in BioBank Japan. This process will be in the web base, and it will take only a week or so before approval. Therefore, there are very much quick procedure for those steps. Though we do not offer individual sample data distribution service to overseas currently, but we hope the near future the service will be available to overseas researchers. There are several tips. There are using sample genetic data in our project. Recently, there are quite a few good results obtained. For example, 6 new genetic loci were identified to be associated with atrial fibrillation. Also, 28 new genetic loci were identified to be associated with type 2 diabetes. Then the genetic risk score were identified for coronary artery disease. In addition, recently we have the several interesting research results shown here. For example, large-scale DIVA study of 42 diseases was performed by using BBJ data, that is the non-European individuals. This study provided multiple insight into the etiology of complex trait by integrating annotation of missense variants, EQTL variant, and transcription factor binding site. Also, polygenic risk score associated with lifespan were elucidated. Furthermore, chromosomal mosaicism was identified in age-related hematopoietic clones and diseases. However, there are several tips towards a better biobank for research. First, follow-up clinical data collection and database is very much important. Initially, we collected clinical data from the paper-based clinical record, but based on the electrical health record is very much important. And also, further genetic genomic study will be important. We finished all of the individuals were analyzed for the SNP RNA analysis, but now we are going to have more and more results by the whole genome sequencing. And in the thirdly, we'd like to have a multi-omic study, including metabolomic and proteomic and other analysis to implement for the good research. For the first step of the issues, we try to establish a system, electronic health record-based clinical database. By you collaborated with the more than 20 hospitals, we collected more than 1.5 million ICD-10 codes for the 120,000 patients in the last year, the category of the ICD code. Also, we got the clinical database based on the EHR. As you see here, we have 73 diseases ICD-10 code with more than 5,000 individuals. And furthermore, as I said earlier, follow-up and genomic data will be very much important. Since the genomic and multi-omic data is important to clarify biological mechanisms of diseases, as well as to predict disease outcome and find out the prevention of disease progression. Therefore, in the past, we have the clinical data, lab data, as well as the SNP array results as a resource for the researchers, but we have to get more results or analyze the results of proteome and the metabolome, like the for the omics data, as well as the whole genome sequence data. So this part of the information we are now trying to get accordingly. So we have some of the progression of metabolomic and genomic study based on the NMR analysis. 10,000 BBJ sample results used, and we got a fairly preliminary but very good omega-3 fatty acid GBAS result. And also, ongoing proteomic study since methodological progress enables to obtain large numbers of protein study results. Therefore, we are now preparing to study the 3,000 samples for proteomic analysis as a trial with the already performed whole genome sequence results. So this is the main major researchers for our project. Thank you very much for your attention.